Welcome to the Virtual Roadshow series presented by Channelchek and Noble Capital Markets. Of the 6,000 small and microcap companies listed on Channelchek, today we're featuring Dyadic International, NASDAQ ticker symbol DYAI. Research coverage of Dyadic International is provided by Noble Capital Markets. Noble is a FINRA-licensed, SEC-registered broker-dealer. All Noble research is available on Channelchek. Following the formal presentation, Ahu Demir, Noble's biotechnology research analyst covering Dyadic International, will ask our presenter a selection of the questions received during the broadcast. And now, here is Mark Emalfarb, President and CEO of Dyadic International, to make the presentation. Mark, take it away.
Thank you. Good afternoon, everybody. I want to start out and just point out that we're working on solving today's problems with the pandemic, solving tomorrow's potential future pandemics, epidemics, potential biological outbreaks. And then most importantly, we're working continuously on developing the technology and applying it for animal and human health for both vaccines and drugs for farm animals, chickens, pigs, and cattle. In addition, of course, dogs and cats, companion animals. And then on the pharmaceutical space, we continue to work with major players. But I want to, as we stay on this slide, and we'll jump to the next one, is the highlights here is we're very flush with cash. We had $32.1 million in the first June 30th after the first half of the year. We went through about $4 million. So we have many, many years of running room here. And we expect to get license deals with upfront cash at some point in the not too distant future, one or more of those, maybe Q1, Q2, potentially this year, more likely next year. That could take us really with multiple years in addition cash, potentially even decades if we can get some of the upfront cash we would expect. We have no debt. I think you realize that. There's no warrants. So we have a very clean cap table. And of course, management, including myself and my family, own a significant portion of the business. So we're in it with you all the way till we really hit the home run of the grand slams or multiple grand slams on vaccines and drugs for both human and animal health. It's an interesting sort of setup. We've set up a virtual business here, which allows us to speed the development, lower the cost, to do things we otherwise couldn't just do on our own, because we're using experts in the field to do the exact same things we need to do. But I want to remind you that we have over 20 years experience in biotech, and you'll see that with the management team. But more importantly, we've spent 2.5 decades prior to jumping in only in the last few years in the pharmaceutical industry, building this cell line so that it can produce massive amounts of low-cost proteins at flexible commercial scales. And we've produced enzymes at scales that we're never going to need in a pandemic or otherwise. So C1 really is here at the right time, in the right place in history, and we expect and are trying to apply it to make history and save lives globally all over the planet. The leadership team is very lean, very mean, but very focused, and we have experts in the field. Dr. Bose, in fact, was just on a call this morning with us. These people are very active, our board members, with a major pharmaceutical company. We had a conference call this morning to talk about where and how potentially we can apply C1 to an area that the world really needs, and I think we can help fulfill the void. Dr. Buckland, also 29 years at Merck, very experienced, more on the vaccine side than the antibody side, where Dr. Bose is more on the antibody side, but they cross over. And of course, with Michael Tarnock, vast experience, corporate governance on the chairman of the board. And the management team, let me tell you, I couldn't have a group of better people to work with. It's 24/7, 7 days a week, late at night, Sundays, Saturday, doesn't really matter. That these people are committed to win. They're committed to drive this across the goal line for a lot of reasons. And I think I was on a conference yesterday as a panel member, and it's really the whole pharmaceutical biotech industry has put their heart and soul into really attacking this pandemic and solving the problems we're all facing today to try to get life back to normal. I think C1 and Dyadic is at the core of that, because without the ability to mass produce, whether it be a vaccine or an antibody, we're not getting anywhere soon, and at the cost that the world can afford. And so I think we have a great great opportunity not only for the pandemic to help, but really to help future and drive the cost of drugs down long term and make it affordable healthcare to people all over the planet. So what are we doing and why are we doing what we're doing? So first and foremost, I'll skip to the bottom, the internally funded programs. If we don't modify the cell line, which is what we've been doing really for the last 4 years, so we knocked out proteases, that allowed us to make this receptor binding domain at now 3 grams per liter. And I don't think people really understand, the investors, what that means. That meant a month ago, we were only 1 gram per liter, which was already record levels that allowed us to produce billions of doses quickly and affordably. We can now do that at 3 times the level. So that's amazing. I mean, if you really think about it, and the industry's looking at that and they're shaking their head, how's it possible? And it's possible because we spent 25 years creating this to pump out massive amounts of protein in the industrial side. And what we've been telling you in the industry is we've been modifying this to address a serious problem that's now coming to surface with the pandemic. The manufacturing of drugs, vaccines, it's inefficient, it's slow, and it's too expensive. And so we're going to change that. We're going to disrupt that with this cell line on multiple proteins, multiple vaccines and antigens and therapeutic drugs. And then the glycoengineering work we've been doing, that's focused on monoclonal antibodies. And you need human-like glycans for some things and other things you don't. And we're on our way to developing, and we have cells that we have developed for human-like glycans. And now, so we need to combine the hyperproductivity of one cell line, the protease deletions of another one, and the glycoengineering of a few of the ones of the G0, G0F, G2, and put those all together. And once we have that, we'll have, we believe, the complete picture to the puzzle. But in the meantime, we can make things without the complete picture. For example, the antigens for these infectious diseases, animal health, etc. We're making those at record levels, very low cost, in significant quantities that's going to make a difference. So that's what we're doing with our internal money. We also worked on some metabolites. We'll focus on that sometime in the not too near future. And then we also, if you think about external programs, what are we doing here? We're applying their money, their drugs, their potential candidates, and we're learning. They're paying for some of this R&D development, the improvement of the cell line, targeted towards their specific proteins. We're knocking out proteases on antigens and other proteins that are potentially commercial targets for some of these big pharma companies. And when I say big pharma company, we're talking to and have several in and more coming that are the biggest ones on the planet. Top 5, top 10, top 25, top 50, small biotechs. I think, you think about it, and the business model here is we've got them in the door, we're creating opportunity, and we're in discussions with 1 or 2 of them for potential opportunities to take licenses, which could contain eventually upfront access fees, milestones and royalties, and then potentially even broader, bigger opportunities for us and for their customers and patients globally. And then there's smaller biotechs where we can get equity investments, funded sometimes research if they have the cash, milestones and royalties, or partnerships. And then there's governmental grants and agencies. We're working with, of course, the IIBR, the Israeli Institute in Biological Research, and the Frederick National Lab is still going forward on their vaccine candidates, and that program is moving in the right direction, and we hope to be able to report positive news on that in the not too distant future as well. So what are we doing here? We're applying their cash along with our cash to exponentially advance this technology without burning a lot of money and being fiscally responsible. So people ask us, well, what is C1? Well, bottom line is C1 is something that can be used to improve the manufacturing process. It's something that could be used to do things faster, cheaper, in larger quantities. And with the pandemic, it's there at the right time at the right place in history because we need a lot of vaccines produced and monoclonal antibodies, which we'll talk about in the slides coming up. It's fast, it's robust, it's versatile, it's programmable, it's scalable. It has everything you need. So it's just a matter of now perfecting some of the little things that need to be improved on, like knocking out the proteases, which we've done to a great degree, the glycoengineering, which is in progress, and then applying it all together to solve hundreds, if not thousands, of opportunities to apply this technology. So competitive edge, what is it? It's about purity, it's about productivity. There's no endotoxins, for example, like E. coli. So let's put it head to head against some of the things that we're competing with. In CHO, we can do things quicker. We can produce antibodies, 3 or 4 batches of antibodies you'll see coming up, in the same time as one CHO produced batch, potentially with a higher productivity per each batch on top of it and a lower cost of food. It's the unique morphology that makes the difference here. That serendipitous mutation that happened decades ago that we've exploited, and we've applied these genetic tools and the synthetic biology too to make it robust and versatile with the wide pH and temperature ranges, which allow us to do things that you otherwise couldn't do with other organisms. And it's really the statement, and I've been saying this for over a year, maybe 2 years to you guys, it's about the science. And the data doesn't lie. The data says what it is, good, bad, or indifferent. And you know, in the ZAPI program, we did both the SPV and our VFV. We expressed them at very high levels. One example, SPV, 300 times more than baculovirus, which happens to be the cell line Novavax is using to make their SARS-CoV-2 vaccine candidate. It happens to be what Sanofi's using. And they're using that not because it's better. In fact, it's far inferior. It's because they had it, they were used to it, they were comfortable. There's really no really rational reason why they're using that over C1. So we're going to hopefully in not too distant future just bypass and blow by with the race in terms of productivity. There's no comparison between C1 and baculovirus cells. And that was shown again in the Sanofi flu vaccine program, which is on our media center presentations. If you scroll all the way down, you'll see that in the case of Sanofi, not only did we produce more, but it worked better, gave a better immunogenic protection using 1/3 the amount of the protein. When you combine that all together, we think we have a revolutionary way to develop and produce vaccines. Next generation biomanufacturing, quick, affordable, all the things we talk about. Again, here it is, fast. We won't go over some of the same things on this particular slide. So here we go, let's talk about it. I think you should be amazed at the fact that we actually have 7 different collaborations going on and up to 10 animal studies already that should be ready by the end of the year, the data coming in. We already have data coming on hamsters, on mice, on transgenic mice with the ACE2. So we've got everything going, the pieces of the puzzle on the animal studies that we need to do. Now we need to get into human clinical trials. And we're moving forward on that on a couple of fronts. And we hope to be able to do that, not using our own money, but other people's money. But if we have to, more likely than not, we will use our own money. And we'll do that sometime no later than Q1 2021. And the IIBR results, efficacy, immunogenicity, titers, neutralizing antibody look very, very promising. And we expect more and more substantial, efficacious, and safe data to guide us in the future of what's the best way to apply this technology as a vaccine candidate. And you'll see also coming up as not only vaccines, but potentially use it for antibodies as well. And so this slide it says antibody ready. Well, you know what? We've been antibody ready to some degree for over a year. We have antibodies we produced, as you know, at 24 grams per liter in 7 days. That's another record level. And that was not even optimized. As we just talked about within the last month, with about 4 to 5 weeks of fermentation optimization on the antigen vaccine candidate, we went from 1 gram per liter, which was already a record level, to 3. So now we're at record level. So we can push these further. We don't need to at the moment because we're working on quality issues, but there's a potential to apply this right now as it exists without any further glycoengineering, potentially either in the G2 strain, the G0 strain, or even the original Man3 to Man9 strain, because these antibodies bind and neutralize virtually identical. And most, if not all of these MABs, monoclonal antibodies for SARS-CoV-2 cocktails, like the Regeneron cocktail, you just need to bind and neutralize. These aren't things that are going to be going long-term in patients over a year, 2 years, like the other antibodies. So we can come in potentially quick. If there is some immunogenic issues still there, hopefully they won't cause a problem. And then as we glycoengineer it further, they'll be eliminated. So we're going to try to apply this now. And as we mentioned the other day in our press release, we've already expressed a SARS-CoV-2 antibody with a small biotech company that's very promising. So we're going to see very soon what the yield was of that, what the quality is, and provide that sample back to them They can compare that to CHO-produced antibodies that they already have done and see where C1 stands. And we're in discussions now and talks and conversations with other big pharma companies about how we're going to solve the problem. Because no matter what, nobody can make enough antibodies for the SARS-CoV-2 treatments, the cocktail antibody treatments like Regeneron or Lilly. And we think C1 can supplement that. We're not telling them to replace it. It's a great opportunity because you know what? They're not going to be able to make enough. And so now we're saying, keep doing what you're doing. Let's just supplement it with this and have additional antibodies and treatments available to save lives globally. And then again, this is just a recap, enormous productivity. That RBD used to be 2,200. Now we have an example with this new antigen that we just produced for SARS-CoV-2. It's 3,000 milligrams per liter. If you remember, and I don't know what the baculovirus number is for their SARS-CoV-2 candidate, I could speculate, but in the ZAPI program was 6 micrograms per liter. We were 300 times more. So we don't need to be 300 times more. If we're 10, 50, 100, I'm sure that's more than enough. And so we're going to apply that technology and are doing so in a variety of programs, including potentially with the new announcement the other day, yesterday, and with Epigen, which has the infrastructure and facility to produce microbial fermentations all the way from the cell line that we deliver to a vial that can be shot in a person's arm. And so that's an exciting opportunity. And I'd say stay tuned because we have a variety of other ones that are incubating that we're promising and hopeful that we can bring out good news from those as well. So low risk, high reward, plug and play. We can develop cell lines very quickly. So if this gene shifts and we find out, oh my God, the vaccines we're making today, we now have a shift in the virus and we need to come up with a new sequence. Guess what? Today, C1 is better known because of the pandemic, because of the opportunities, because of the people we're working with, because of the people we're not working with, but we're in discussions with, that they potentially take the new gene sequences for antibodies and vaccines and put them into C1. So that could be exponential new clients for us today and tomorrow. And then of course, we have multiple shots on goals. Here, if we just get into man, prove safety and efficacy, that's going to open up the doors for C1, we believe, very widely and very deeply to be able to allow us to do animal and human health at an exponential rate in candidates. And again, you know, we've expressed the RBD at enormous levels, and you can bet that people like the Frederick National Lab and other people are seeing positive results for different candidates than the RBD that could be potential vaccine for the SARS-CoV-2 or other infectious diseases in the future. And we're working on 4 different continents right now. So things are moving in the right direction, that we can tech transfer this technology very quickly. Within a month, the Indians will be able to be producing C1 from this technology. Will they get 3 grams per liter off the bat? Maybe, maybe not. But if they only get 1 or 2, and then in a month or 2 down the road, as they continue to improve it, as they hopefully get the funding to bring those clinical trials to bring this to the phase 3, ultimately in the Indian continent, guess what? Even 1 gram per liter is enormous amount, but we'll ultimately get 3 or more in India. And just like the Israelis, the tech transfer goes very smoothly. We've done it many times. So the key takeaways here are next generation platform. It's not new, it's been tweaked. It's been 2.5 decades under development. It's been used commercially, it's been used industrially. BASF, Shell Oil, Abengoa Bioenergy. DuPont paid us $75 million to get access to our industrial business, including the cell line for the industrial business. It's disruptive. We think we can just change the game. We're partnering it already with top tier animal, all 4 animal health companies are funding right now, in addition to Fifth. We're in discussions with others, multiple parties on the human side. And guess what? Some of the people that were in the door left are coming back. Some of those people are talking to us about potentially getting a license. That license may be today or after funding, but they now are talking about terms that go with a potential drug that if we're successful and they want to bring it forward, they now want to know what it's going to cost. And again, we have a highly energized management team and plenty of cash to execute our plan with no debt. And with that, I'll take it back to Ahu for questions.
Thank you, Mark. I am Ahu Demir, the Noble Research Analyst who covers Dyadic. Here are the few questions I have selected, as well as a couple of my own. So the first question, Mark, is, as it's the major topic of today's world, and you recently provided an update, can you elaborate on the coronavirus programs and partnerships?
Yeah, so we have 7 different collaborations going between governments, academics, commercial parties, including biotech companies, and they're evaluating C1 in vitro and in the animal studies, and then determining if they want to move those forward, what it's going to take, what doses they get, what would have to be required. But the beautiful thing here about C1 is we're approaching it from 3 different directions. We can make the RBD just by itself, and we're probably the only ones that have the luxury to do that with not even an adjuvant, which for elderly people may be actually a safer, effective vaccine. We'll see that in one of the animal trials. We're working on that, maybe 2. We're using the RBD with an adjuvant all by itself. It's a simple, easy way to produce a large volume of low-cost vaccines, potentially, for example, the Indian continent. And then the third one is we've also produced the RBD with a spike tag that will allow us to couple it to a nanoparticle and do the same exact thing that other people are doing for nanoparticles, which may be more effective, or you can do it, but it's more costly. So we have these 3 different approaches, and in some of the animal studies, we're trying to determine which is the best approach from a safety, efficacy, and a cost of goods. And it may be that one party wants to go like an Indian continent for low cost effective. Another one may want to go for a little higher cost, a little more effective. But the beautiful thing here is we have these options because of the hyperproductivity that others don't have. And then in addition to just what's going on with our own candidate, and that is our own candidate, you may not understand, most investors, the gene sequence in that cell, this is a dyadic vaccine candidate. All 7 of those programs are using our candidate, not their own. And then we have, of course, a candidate or candidates, for example, with the Frederick National Lab and the NIH and NIAID, where they provided their gene sequences, and that program's still moving forward. It's looking promising. Where that's going to go, I don't know, but the science looks good, the data is starting to look good, and we've done that fairly rapidly. So hopefully that gives you an update as to what's going on there.
That's great. And if we would move to Frederick National Lab partnership, how was the partnership was established? And also, what are the basic fundamentals of the projects and potential timelines on that partnership?
Yeah, well, that's the U.S. government, so the timelines, who knows? But I would say that, we have a great communication, we have great respect for one another. Things are going very well. I think we're demonstrating that we can produce that product, that candidate that they have. I think that they're excited about it. I think they're narrowing it down to what they want to use and potentially apply into hopefully animal studies and clinical trials and ultimately commercialization. And the beautiful thing there is not only are we proving ourselves in this particular context, but like we did in ZAPI, will we hopefully become embedded in the future opportunities as well. And of course, they're working on normal traditional drugs as well. But the other thing here is the antibody opportunity for C1 is huge. It's always been huge. It's hundreds of billions of dollars of prescription drugs. You should focus on the fact that Scott Gottlieb had made a statement a couple of years ago that 2% of the American public has access to biologics, and it was 40% of our prescription drug budget. With COVID, nobody can afford to continue doing the same things over and over again and expect different results. So ultimately, we're going to crack that egg, and it's going to open up and things we believe are going to be exponential growth for our technology platform to be used and applied broadly on the planet.
Okay, and if you would move beyond coronavirus on your portfolio, you have such a rich partnership portfolio. Can you give some information on the current partner research projects?
Yeah, I mean, I can't give you specific details, but we are in discussions now with 2 different parties. Both of them are big pharma companies that are talking about potential license terms. Now, that doesn't mean they're going to give us a license up front with cash immediately. That means they want to know when they put more money in because they see good progress and they're excited about the opportunity, is what it's going to cost them in the end. And so we're beginning those discussions now with 2 parties on that's human health. And then animal health, we're hoping to get one of these or more of the animal health proteins into an animal study towards commercialization. And then, of course, we have multiple new people coming in all the time, in addition to expanding what we're doing. And we're still waiting for Sanofi, by the way. So I think they've been preoccupied with their own COVID programs, but we're still waiting for them to come in, and hopefully they're going to pick one or more molecules that they want to apply C1 to, and we can move that forward as well. So there's a lot going on, and I would say that we're sort of a victim of riches because things are getting very, very busy. They've been very busy for the last 3, 4, 5 months on COVID, but on top of that, it's opening the doors up and their eyes are getting wide open. So if you look at my eyes, that's what's happening. They're recognizing the power of what we have over. A lot of times we get the questions, why isn't everybody using this program if it's so powerful? And we ask the same question, why? Pharma's conservative, it takes slow, their time, but we're breaking those barriers down one by one by one. And things are moving. So we see that instead of swimming upstream against a huge sort of flow of water, it's slowing down and it's getting easier to make these conversations turn into meaningful opportunities.
Okay, so the follow-up question would be, how do you plan to monetize all these possibilities?
Well, that's a good question because it's a fluid plan. Of course, we expect, as you go back to the first slide, what we did on the industrial side, we're starting to repeat on the pharma side. Non-exclusive licenses with upfront cash payments, milestones, and royalties. And if you remember, we expect magnitude times upfront cash payments for biopharmaceuticals than we got for enzymes. In that case, we got $30 million from the likes of Shell, BASF, Abengoa upfront. Then we would have got milestones and royalties. And of course, DuPont bought that out for $75 million in the industrial side. So that's one way. Two is there are potentially joint venture opportunities where we can be partners, where we don't need the cash with small biotechs or even big pharma. They have things where we've done programs, for example, we might have hit scientifically the result and got the benefit of it, but they're now not interested in it as a company. But for us, it could be a huge opportunity. So we have one or more of those molecules right now that we're trying to think about how do we out-license those technologies, get them out of the pharma company where we did a great job, proved the potential benefit of hyperproductivity and lowering costs of a multi-billion-dollar drug, and move that forward without us spending our money. Or we may take the model in time, if we get some cash up front from a license fee, of doing things for animal studies in phase 1 on our own, and then out licensing them to partners. So there's a variety of opportunities to cash in, so to speak.
So the next question is more focused or specialized on the partnerships. Can you update us on the progress of evaluation of WuXi assessing C1, and also when will we hear about the Sanofi's decision to move forward?
Yeah, so starting with WuXi, we've transferred the technology to WuXi. We're working on, there's several different genes that they're testing it out with. And so it's in their hands. They're starting to do the genetic engineering to do the strains and to grow it up. And we're talking to WuXi about also having them access some of their customers with some of the opportunities. Because of course, we'd like to be able to deliver 1 billion doses into China of an affordable, very safe, effective vaccine. And what you got to realize is this vaccine can be used as a boost. Not only can it be used as a prime and a boost, but it could be a boost to the adeno vaccines, the mRNA, the DNA vaccines that are struggling in problems, not only potentially with safety, but also with efficacy. We think that they're going to need boosts, and we think we could be that boost. And to be honest with you, the receptor binding domain approach that we took, that came from deep thought, from the experience, from the European scientists, the 3 different guys that were in the SARS and MERS and COVID, top 20 scientists, Tho University, Utrecht, Erasmus. These guys knew from their experience that the RBD was a safer approach, not having ADE, so we have less chance of safety. We can make it in a, let's say, higher yield because it was a smaller fragment and doesn't have all the protease sites that the spike protein does. So there was a lot of thought going into this. And then also with the nanoparticle, that it's more effective, you can use a lower dose. The beautiful thing here is we believe that we can get away without using nanoparticle by using a higher dose. And because we make so much of the RBD so fast and so affordable, we might not have to get into a complicated manufacturing process. We've licked the mass production that everyone else has struggled with. Now we just need to get in the man, prove it out, safety, efficacy, and virtually use this technology platform for generations to come.
So if you would move to the next question, throughout your presentation, you mentioned potential initiation of human trials in the first quarter of 2021 via partnership or company sponsorship. Which projects, modalities, or indications you plan to advance into the clinic?
Well, the SARS-CoV-2 vaccine is the closest thing, but the biggest impact right now. And that's the one that the company, at least we have control over. That's, remember, and I don't think investors get this, that's our vaccine candidate being used by others. We're kind of like an API supplier. If you look at this, is we can either make the cGMP material and provide that to multiple parties and just sell it as an API, the active ingredient, and then they can formulate it however they want. Or we could license the technology and let them grow it up and produce it like we're doing with the Indians on the Indian continent. They happen to have their own facility. They have the ability to put it into vials, so it makes a lot of sense. So there's both approaches, but that's our vaccine candidate and we have control. We don't have control over what pharmaceutical companies want to do on their own. So that's the most likely thing that's going to go in, but there are other products potentially could go in from one of our project partners.
Okay, and if we switch gears to animal health, the path of approval for animal vaccines and therapeutics is quicker than for humans. So one question we received was, considering companies are testing C1 for a few years, animal companies, why is the commercialization is taking longer than expected?
Well, first of all, only one company has been testing it for over a year. And we have 2 or 3 proteins we worked on, and that's one of the ones we hope to get in. Timelines, again, these aren't things we control in that regards. And the other ones, the other top 3 or the top 4 are all somewhere like a month and a half ago, somewhere 6 months ago. So, those are programs moving forward, but in some cases, they either will or won't go forward, but it's demonstrating the power of the technology they may want to bring in-house and use broadly. So a lot of these programs are really proof of concepts. Some of them are moving towards potential commercialization, but the proof of concepts that they could bring in or now decide we're going to adopt C1 to use for certain classes and types of molecules, and those classes and types may expand as we further glycoengineer the cell lines and modify them to fit exactly their needs.
So my last question, Mark, is on, as you implement an efficient business model with these partnerships, can you tell us about the cash runway and burn rate, and when do you think the company will become profitable?
I think I'm a broken record. So we have plenty of cash. I want people to just think about it. We own a big chunk, so we're watching the cash like it's our cash. Your cash and our cash is the same thing. So we own about 30% of the cash. So we're very, very careful with that. And we have no intention of diluting shareholders for unless there's a huge reason and an opportunity that we just can't pass up. So it doesn't mean we're never going to do that. It means we're going to do it very cautiously if we decide to do it at all. If we get a license agreement in with an upfront cash, doesn't mean we're going to raise money or use an ATM. It means it's there for the opportunity if and when we seize it and need it. We don't need it right now. Right now, what we need to do is do exactly what we're doing. Improve the science using our money to do it, but leveraging third-party money to do it. Because I want you to understand, when we knocked out 14 different proteases, we learned which proteases to knock out based on antigens, monoclonal antibodies, therapeutic proteins that came in from our partners. We didn't have all these drug candidates or vaccine candidates to know which proteases to knock out. That's invaluable, not just the cash. And then we give them back the protein and they do analytics. And we find out this worked perfect, this needs to be tweaked over this way, tweaked that way, and we go back and we modify it. So that it's not just the cash, it's the data, it's the access, the partnership, And some of these partnerships are growing. And as I mentioned, there's 2 of them right now commercially that we're in discussions, and they want to have further discussions about terms. At the beginning, these guys just threw money, proof of concept, let's see what it does. Now we've proven enough that they're not comfortable, because guess what? Somebody can come in and this thing can go away tomorrow, or a portion of it can go away tomorrow. So I don't know if that answers your question, but I wouldn't really worry about our cash right now. I'm not worried about it, and it's 30% of it's mine. So you shouldn't worry about it.
Well, Mark, that's all the time we have today. Thank you for helping us get a better understanding of the company's story. If you would like to get the independent research I have written on Dyadic, go to channelchek.com and type the company name or the ticker DYAI in the research box located in the top left company data section of the site. Again, thanks to our guest, Mark Emalfarb from Dyadic International.
Thank you, everybody, and stay safe, because we're trying to keep it that way.
This has been the Virtual Roadshow series brought to you by Channelchek and Noble Capital Markets. Log into Channelchek or subscribe to our YouTube channel to get the full catalog of roadshows and C-suite interview channelcasts. Register for Channelchek's no-cost premium content and receive notifications of new releases from both series. Thank you for joining us.